Welcome you all. Myself, Miss V.B. Mani, working as Assistant Professor at Satara College of Pharmacy, Satara. In this video, we are going to see how to determine maximum wavelength of paracetamol sample. So the aim of the experiment is to determine maximum wavelength of the given paracetamol sample by using double beam UV spectrophotometer. After performing this experiment, these three types of the objective will be achieved. So the first one, student will understand the concept of lambda max. Second one, student will acquire the skill of handling double beam UV spectrophotometer. And the third one, student will understand about how to prepare different dilutions. Now, these are the requirements, this type of the glasswares, instruments and the chemical which will be required while performing this experiment. Now we have to see the principle involved in the determination of the lambda max by using UV spectrophotometer. So, first of all, we should know the meaning of wavelength and the maximum wavelength. Wavelength is the distance between two successive maxima or minima, and lambda max is the wavelength at which maximum absorption of the electromagnetic radiations takes place by molecule due to its electronic structure. So, in this figure, this is the peak point, that is here at this point maximum absorption takes place. So, at which wavelength the maximum absorption takes place? So, at this wavelength the maximum absorption takes place. So, this is the lambda max. The basic principle of UV visible spectroscopy is based on the absorption of UV light or visible light by chemical compounds, which results in the productions of distinct spectra. Spectroscopy is based on the interaction between the light and matter. When the matter absorbs the light, it undergoes excitation and de-excitations, resulting in the production of spectra. When UV or visible radiations are allowed to pass through a medium or a matter, it absorbs the UV radiations. Energy of the electron present in the molecule undergoes excitation. This causes them to jump from a ground state energy level to an excited state energy level. Small amount of the energy associated with ground state, whereas large amount of the energy associated with excited state. It is important to note that absorption will occur only when the energy of the exciting photon or energy of the UV radiations matches to the energy difference between the ground state and the excited state of the absorbing species. These energy difference are different for the different species. Absorption of an electromagnetic radiation in the visible or the UV region by molecule is due to the presence of the chromophore in the absorbing molecules. So, chromophore is a covalently unsaturated group responsible for absorption of electromagnetic radiation in the visible or UV rays. It may or may not impart any color to the compounds. The examples of the chromophores are carbonyl, alkene, nitro group, alkyne, etc. Now, we have to see the procedure for the preparation of the pure drug solution. Here the paracetamol pure drug sample will require for the preparation of solution. Step by step we are going to see here. So first step here, weigh accurately 50 milligram of the pure paracetamol powder. Then transfer this measured amount of the paracetamol powder into 50 ml capacity of volumetric flask. Then pipette out 12 ml of the prepared 0.1 normal sodium hydroxide solution and then add or transfer into 50 ml capacity of the volumetric flask and dilute the solution up to the 50 ml with distilled water. So this solution is the standard stock solution. You have to make the label on that volumetric flask to avoid the confusion. Now, dilution number 1 solution is prepared by using the standard stock solution. So the strength of the standard stock solution is 1000 microgram per ml solution. So for the preparation of the dilution number 1 solution, pipette out 5 ml from the standard stock solution and transfer into the another 50 ml capacity of the volumetric flask and then dilute up to the 50 ml with distilled water. So when you calculate the strength, you will get the 100 microgram per ml solution. So this is the dilution number 1. Now, prepare the final dilution by using the dilution number 1. Pipette out 5 ml from the dilution number 1 solution and then transfer into the 50 ml capacity of the volumetric flask. Then add 5 ml of the 0.1 normal NaOH solution and dilute up to the 50 ml with distilled water. So you will get the strength of the solution is 10 microgram per ml. You have to make the label on that volumetric flask as a final dilution. Now, similarly, we have to prepare the blank solution. Whatever type of the solvent which is used for the preparation of the final solution, same type of the solvent we have to take as a blank solution here. So, in the final dilutions, we have used 0.1 normal NaOH solution and distilled water. So, similarly, we are going to prepare here the blank solution. Now, pipette out 5 ml of prepared 0.1 normal sodium hydroxide solution, transfer into the beaker and dilute up to the 50 ml with distilled water and make the label on that volumetric flask as a blank solution. Then record the spectra of the final dilution in the spectrum mode in the wavelength region 200 to the 400 nanometer and determine the wavelength of maximum absorption. So this is the point where the maximum absorption takes place, but at which wavelength the maximum absorption takes place, that is at this wavelength. So you have to calculate this one. You have to determine at what wavelength the maximum absorptions of the electromagnetic radiation takes place from this spectra. Now this is the observation table. Here in the second table, second column, name of the sample you have to write here. Then you have to write here the concentration of the final dilution, that is 10 microgram per ml. Observed lambda max. After getting the spectra by UV spectrophotometer, you have to determine at what wavelength maximum absorption takes place and note down here and write here. And you have to write here the absorptions of the solution. So both these data you will get from the UV spectrophotometer after recording spectra. And then finally you have to write down the results and the conclusion. So these are the references. Thank you all of you.